Hello everyone, I'm Dr. Arpita. A very warm welcome to my plant physiology class. The topic for today's discussion is phloem translocation. This basic concept of phloem translocation is already known to us. We know that food is produced in the source region of the plant, that is in the leaves, and it is translocated. The photoassimilates are translocated to the sink location or the roots or storage area. Correct? So this movement of photoassimilate or the products of photosynthesis in the phloem is known as translocation. Right? Now this picture, I think you are familiar with. You know what is this picture all about. This is about phloem tissue. You know it, correct? Phloem tissue consists of, it includes sieve elements, fundamental phloem, and companion cells. Correct? These three things you already know. They are made up of sieve elements, then they are having fundamental phloem cell, fundamental phloem cell, and companion cells. Right? Companion cell in angiosperm and analogous to this is albuminous cells which are present in gymnosperm. Correct? And the sieve elements will have what? Should know this. Sieve elements have sieve tubes and sieve cells. Sieve cells. This is something you already know. Clear? Now our entire discussion will be based on the movement of the photoassimilate from the source to the sink through these elements, these components of phloem tissue. Clear? Now see, companion cells are of three types again. The ordinary companion cells, the transfer cells, and intermediary cells. This again is of three types. What are they? The ordinary ones, ordinary companion cells, transfer cells, and intermediary. Intermediary cells. These ordinary companion cells, they normally have a smooth wall with few or no plasmodesmata. That means they don't have plasmodesmata connections, or maybe in some cases they have very few connections to cells other than sieve tube. Fine. The transfer cells are like the ordinary companion cells, but they have much folded walls that are adjacent. Okay, that are adjacent to the, see, understand the concept very nicely. Okay. So what did I say? They have much folded walls that are adjacent to non-sieve cells, allowing for larger areas of transfer. That is why they are known as transfer cells. That means they allow a greater area for transfer. And last, is the intermediary cell. These intermediary cells have smooth walls and numerous plasmodesmata connections that connect these cells to each other. Fine. These transfer cells, they transfer sugar from the apoplast to symplast of the elements and companion cells in the source. On the other hand, what do these intermediary cells do? They function in the symplastic transport of sugar. So these two keywords you remember, they perform their function in the apoplast region, and intermediary ones, they do symplastic transport. Correct? So one is doing apoplastic transport, the other is doing symplastic transport. Symplastic transport of sugars from the mesophyll cells to sieve elements. Clear? Okay. Now, we'll talk about the mechanism. Fine. Now, before talking about the mechanism, I would like to ask you a question. You must be knowing that glucose is formed in the form of photoassimilates in the plants, leaves, right? But this glucose is not transported to the sink. Why is it so? Have you ever given it a thought? The material that is translocated in the phloem is not glucose. It is sucrose. The reason is very simple. The glucose that is formed is reactive. Glucose is reactive. It is reducing in nature, whereas sucrose is non-reducing sugar. It doesn't react. Sucrose is translocated to the sink. Fine. So sucrose is the most commonly translocated sugars. And generally, the translocated carbohydrates are mostly non-reducing in nature. And sucrose, you know, it's a non-reducing disaccharide that is made up of one glucose and one fructose. Clear? Fine. Now let's see what happens. But before going to that, I would just like to make this concept a little clear to you. What is apoplast and what is symplast or apoplastic pathway, symplastic pathway, all these, because we are going to discuss about this in the later part of our lecture. In case you have forgotten or you are not being able to recollect what is apoplast and what is symplast, just take a look at it. You'll quickly be able to recall. So loading actually happens through two pathways. If I write here, see. See here, loading happens through two pathways. Apoplastic pathway and symplastic pathway. In apoplastic pathway, what is happening? It is through the cell wall. And symplastic pathway is, what is that? Through the cytoplasm, connected via plasmodesmata. right? Look at this picture nicely. Look at this color code. This orange line or orange arrow is your apoplastic pathway, and this blue one is the symplastic pathway. See this blue arrow, this part here. See here, this blue arrow is going from here, I mean the cytoplasmic region, and it is traveling through the plasmodesmata and coming to the cytoplasm of the adjacent cell. But what is happening to this orange one? This orange line you see here, it is only traveling, its travel is restricted to the cell wall region and it is not getting into the cytoplasm. This is the cell wall region, this is the cytoplasm, this yellow part, and this is the vacuole. So the apoplastic pathway means it is restricted to the cell wall only, and symplastic pathway is through the cytoplasm. Here are some very important key points about apoplast and symplast. Please take a note at it later. The most important point is the first one. The apoplast pathway is the movement of water through adjacent cell wall, and in this pathway water moves from one cell to another through plasmodesmata. This is your plasmodesmata. This is the connection, the opening, the junction that connects the two adjacent cells. Right? Okay. So now we'll move on to the mechanism. The mechanism of translocation is studied with the help of pressure flow hypothesis. Just let's write. Mechanism of translocation, it was proposed by Munch in 1926. And this is the most accepted mechanism of phloem translocation. It's very simple. Just make it a point to listen carefully and understand the concept. It's not about mugging it up and vomiting out in your paper. That's not the focus. Try to understand what happens here. See, here what it states is that the flow of solution in the sieve element is driven by an osmotically generated gradient between the source and sink tissue. Correct? What did I say? Flow of solution in the sieve element. Flow of solution in the sieve elements is driven by an osmotically generated and osmotically generated pressure gradient from source to sink tissues. To clear? The gradient is it's the consequence of phloem loading at the source and phloem unloading at the sink. So this gradient, Now this gradient is a consequence of what? Phloem loading. Where is loading happening? At the source region and unloading. Where is it happening? This is happening in the sink region. Now see what happens in the in your local retail shops. Every morning or every night, the empty cargo van goes to the factory. It loads all required goods in that, and it comes back in the morning and unloads it in the retail shop. So loading is the procedure where the goods van is getting filled up. That factory is the source from where the luggage van, the goods van is getting filled up with all required goods into it. And where is it coming and unloading? In the retail shop. Clear? So this is the basic mechanism. And here what happens? See, the active loading at the source leads to an accumulation of sugar in the sieve element, which causes a drop in the water potential. The water potential drops in response to the water potential gradient. What will happen now? The water will enter the sieve element osmotically from the nearby xylem elements. In the xylem, the water content is more over there. There is no drop in water potential in the xylem, right? And it will cause the turgor pressure to increase. As water is coming in, it will cause the cells to become turgid, and it will increase the turgor pressure. Clear? See this picture nicely. Very nice observation is required, otherwise you will not understand nicely. More than learning and all, your observation has to be very clear. If your observation is not good, you will understand nothing. Fine? So at source, what did I say? What happens at source? Active loading occurs at the source region. It leads to accumulation of sugars in the sieve elements. Accumulation of sugar in the sieve elements. Sieve elements means what? I have just now told you, sieve tubes, sieve cells, all these. Clear? Okay. Now, this will cause what? This will cause a drop in the water potential. This active loading will cause drop in water potential. This drop in water potential will cause what? Entry of water into sieve elements from xylem. Now see here, this is your xylem, this is the phloem. From this is sucrose, that is the source. See, this is the source region. From the source, sucrose is entering into the sieve element, the sieve tube, and there is a drop in water potential. What will happen? This will cause water from xylem to enter into this. And as a result, what will happen? there will be an increase in turgor pressure. Correct? There will be increase in turgor pressure. This is the basic concept. Now at the sink, what will happen? The sink is what? The end point of translocation pathway. And this phloem unloading, on the other hand, here will reverse, or it will lower the osmotic pressure within the sieve element, which will create a pressure gradient between source and sink, and this osmotically generated pressure gradient leads to bulk or mass flow of materials from source to sink. So according to pressure flow hypothesis, translocation of solute is a passive process. That means what? There is no direct input of metabolic energy, or there is no requirement of input of metabolic energy here. Fine. See, when sucrose moves into the sieve tube, it increases the solute potential. It increases the solute potential there, and water pressure potential in the xylem is very high. Here in this region, see, in this region, the water potential is very high. This part. Water potential is very high. So water will move from xylem into the sieve tubes. Food moves downward by virtue of pressure towards the sink. That's why we say food is transferred in bulk. See, it is numbered as 1, 2, from 1 through 7 here. If you have understood the concept, you can tell what happens in each of these steps. What is happening in 1? See, in step number 1, you can see that sucrose is formed here in the source region. It is coming into the companion cells and it is moving into the phloem elements. Fine. See, these companion cells have large number of mitochondria. It produces the energy via cellular respiration. Clear? Now, this, as I told you, this creates an increase in the solute potential. So, as a result, there is a drop in water potential. What will this do? Here, what did we discuss? It will cause water to move from xylem into the phloem part. See, second step, water is moving out, and third, water is coming in. Fourth step, what will happen? This water will move from this porous plate to the next underlying cells and will finally come to the the sink region. Again, it will go and collect in the sink region, and this happens by virtue of pressure towards the sink. Okay, food is just transferred in bulk, and this translocation of solute is an absolute passive process because no direct input of metabolic energy is required. And here again, what you can see, water is moving out and going back to xylem. At the sink, as sugars are unloaded, water leaves the phloem See, water is leaving the phloem and lowers pressure, and lower pressure results. See, water and its dissolved solutes move by bulk from the area of high pressure. See, I'll write it out for you. You will understand this better. So what happens? Water and its dissolved solutes move by bulk flow from the area of high pressure that is what? The source to the area of low pressure that is sink. Fine. Now these are written here. So in C, step 1, 2, 3, 4, 5, and 6 tells you the same story. I'm not reading it out again. I hope you have understood the concept. I've talked about this earlier also. The dates, don't get confused. Nobody's going to ask you about these dates, but mass flow hypothesis or Munch model, sometimes it is said 1926, sometimes it's said 1930. You don't have to break your head over this point. That's not that important. Just know the concept. I will just give you a schematic diagram to make it very clear to you what is happening in sequence, one after the other, what is actually happening. Fine. So just follow this schematic diagram very nicely. And then I'll ask you one question, which is based on the flowchart. So from here, I'll use two colors. It will help you to understand better. Fine. Okay. See. This is your mesophyll cell. From here, it is the photoassimilate is traveling to bundle sheath cells. From there, it is traveling to phloem parenchyma. Right? These three things are your what? These three things are together your source. This entire thing is your source. Next, it is coming to I'll change the color. It is coming to companion cells. Companion cells. Here, what is happening? Active transport. Active transport happens here. Fine. Now, from companion cells, where will it go? It will go to the sieve tube members. This is, I'll just write here, sieve tube members via pores, or plasmodesmata. This is the pathway of photoassimilate transfer. Now, questions can come from this this part. How you know? You will have to remember the sequence nicely. That are from mesophyll to bundle sheath, then to phloem parenchyma, then to companion cells, then to sieve tubes, and finally it will go to the sink region. Now, these can be jumbled up. These, like, you can be asked to arrange it in sequence. So the source is the first item and sink will be the last, but the middle, in between these intermediary terms should be clear. You should be able to place it in right sequence. You should be able to place it in the right sequence where it should be placed. Clear? This is the basic idea about this phloem translocation. And remember, water transport is unidirectional from the stem, from the root to stem through the xylem. Don't get confused with these two terms, water transport and photoassimilate transport. Please don't get confused. Clear? Still, if you have any confusion, please let me know. See, this before I end, I have told this to you before also, before in the starting part of the lecture, but still I'll tell you one more point. See, in symplastic phloem loading, the photoassimilates translocated into the sieve tube through specialized companion cells. These companion cells are intermediary cells. You have seen these. See, these companion cells are intermediary. These companion cells are intermediary companion cells. These are intermediary companion cells. Clear? By a large number of plasmodesmata, as we have discussed that they are connected through plasmodesmata. But what is happening in apoplastic loading? See, the photoassimilates are first transported into the mesophyll cells via bundle sheath cells to the extracellular apoplastic region, and then by active transport into the specialized companion cells through the transfer cells. So when it is happening through the transfer cells, you remember it is apoplastic and it is an active transport process. So from transfer cells, photoassimilates subsequently enter into the sieve tube elements via the plasmodesmata. Clear, right? The loading of sucrose and amino acids from the apoplast into the sieve elements proceeds via proton symport. This is driven by a proton gradient which is generated via H plus ATPase present in the plasma membrane. Fine. Now again see, there are two possibilities of phloem unloading. In symplastic unloading, what will happen? The source and the sucrose and amino acids reach the cells of the sink organs directly from sieve elements via plasmodesmata, since it is symplastic. In apoplastic loading, the substances are first transported from the sieve tubes to the extracellular apoplastic region and are then taken up into the cells of the sink organ. Please remember this very nicely. Okay, please remember this very, very nicely. So in symplastic loading, what is happening? So, symplastic unloading, what's happening? Sieve elements. Sieve elements are going into the sink via plasmodesmata. And in apoplastic uh, thing, what is happening? The extracellular apoplastic region is coming into play, and it's an active transport process in that loading part. And in unloading, what is happening? The substances are first transported from the sieve tubes to the extracellular apoplastic region and are then taken up into the cells of the sink organs. So this is the basic idea of phloem translocation or translocation of photoassimilates in plant. I hope you have enjoyed the lecture and learned something interesting. If you have found this lecture to be useful, helpful to you, please do share it to your friends also. And forget not to subscribe to my channel and get notified as and when I upload the next video. Till then, thank you.